Afternoon, everyone. My name is Maya Luria with TMC for Seniors, and today we are actually doing something pretty exciting. We've got an in-person audience for today's talk, and Anne is here from TMC for Seniors. She is going to be introducing our speaker today on lengthening the health span, issues of infection and inflammation. You may be watching us on our YouTube channel or our Facebook page today. If you have any questions, please leave them in the comments and we will address them afterwards. Anne, welcome.
Thank you, Maya. Welcome to everyone. This is our first hybrid class. So if I misstep a little bit, please excuse me because we've not done this before. It's the first time we have a live audience as well as people online. So thank you so much for joining us for this class. We're so pleased to have Dr. Janko Nikolich here to speak for us. He is probably the busiest person that I know, and I'm so pleased that he agreed to come and speak to us today. He is head of the Department of Immunobiology at the University of Arizona College of Medicine, and also co-director of the Arizona Center on Aging. And he's got other jobs too, but he's an MD, PhD, someone I like to call an overachiever, because I got a master's degree and stopped right there. So also, lots of degrees. He actually is in charge of the university's department that analyzes and figures out why we age, why infections and inflammation affect us as older adults differently than they do younger adults, and what's happening in our body, what's causing that to happen, and what can we do about it. So fascinating studies that he does. And he's also at this point, I just love this, which is why I realized I wanted to ask him to come to speak. He's now training young researchers in the field of aging. He's gotten money to offer to support them for research for a few years. Usually when researchers start in a field, they stay in that field. So there aren't a whole lot of researchers working in aging. And what we want is new researchers, and lots of them. So at this point, that grant that he got is going to provide lots of new researchers. He's in charge of those researchers and what they study and how they do it. And hopefully we'll have lots of people, because there's different colleges doing this as well. But hopefully we'll have lots of new researchers in aging. It's amazing the kinds of things they study. So I developed a broad title called Lengthening the Health Span, Issues of Infection and Inflammation, because that's really that's the broad aspect of what he studies. And then he's going to pick something to talk about to kind of illustrate that and what they're doing currently in that. So today he's going to talk to us about the immunobiology and aging, lessons learned from SARS-CoV-2. So I've heard him speak about lots of different kinds of infections and things. It's amazing. So I talked to someone on the phone the other day, and she said, what actually is he talking about? We actually hadn't got his PowerPoint yet. And I said, it really doesn't matter. It's going to be great. You're going to learn a lot. So would you, if you don't mind, would you join me in welcoming Dr. Janko Nikolich? Thank you, Janko.
Thank you, Anne. And thank you all for coming. It's, as Anne said, we need to relearn to be in person now.
I know.
We need to relearn that we are persons. That's all important. And then, as Anne mentioned, I will talk about a lot of different things. I will absolutely cover some of what was in the original title. I will cover about some of what's in here. And then at the very end, I will cover the next big adventure that I really want you to hear about, which is by far the most ambitious project that I have gotten involved in. It is called the Aegis Consortium for Pandemic-Free Future. And it comes out of us realizing that we, not as a city, not as a county, not as a state, not as a nation, but actually as a civilization, have flunked this test and flunked it badly. And we have behaved, as one of my favorite documentaries. It's a gorgeous one. I don't know. I saw it, when I was growing up in then Yugoslavia and today's Serbia. It was a U.S. movie about animals, and it was called Those Beautiful Creatures. And it had this one thing where a chimp comes in and picks up this rock and sees a snake under the rock, drops the rock and faints. And then he repeats it 3 more times. And that was us actually with this pandemic. So we feel that we have done many things right in this university, and what we have been able to control by good coordination, but that made us realize what is not around really worldwide anymore. Even what was around is no longer around. You remember the very successful campaigns to eradicate smallpox and to put under control polio and so forth, and we're now far, far worse off than we were. So I'll talk about that at the end. Let's get into this first adventure. These are the people that did the work. We have this adage that when I say I mean we, and when I say we, I mean them, because they did all the work, of course. I kind of like direct and then get out of the way so that I don't mess them up. But, when we think broadly about aging, what can be the goal of an organization that tries to deal with aging? Like the Arizona Center on Aging has these programs that range from very basic molecular work, which is what we do in my lab, all the way to community outreach, to to the hospital and home, living in place, and all telemedicine and so forth. Well, you cannot really say that what do you do? Like you stamp out aging? I mean, you can't. It's not as easy as what you can do if you're a cancer researcher, where you can put a nice catchy phrase. Imagine a world without cancer. We cannot imagine the world without aging. It's really quite unimaginable. One of the important concepts when you think about aging has come about and has been really articulated probably some 15 plus years ago. And it is the concept that's called the geroscience. Of course, when you Google that, it moves you to geoscience, and don't want that. It's geroscience. And it comes from the realization that aging is really the number one risk factor for late life diseases. And if you look at all of these diseases here, diabetes, osteoporosis, vascular dementia, cancer, stroke, you can run the tables and you can run the graphs, and you will see, if you plot the incidence of that disease, how frequently it comes about relative to the timeline, it will go up with age, and it usually takes off somewhere around 50, and it goes like pretty steeply up high. The concept is important because it's telling us that the aging mechanisms, the mechanisms that underlie the basic biology of aging, why our cells and molecules may be growing old in some ways, actually intersect with these chronic diseases. This is a little small, and I apologize for this, but this is basically says that there are some 8 or 9 fundamental pillars of aging, the areas of molecular science that are telling us, based on a lot of evidence, that they are connected to the process of aging. Like metabolism, we know, is very much connected to the process of aging. And if you can kind of like slow down your metabolism or target it in a certain way, there's a very good chance that you might have a very long life. There are experiments done in various animal models where we can deliberately attenuate metabolism, not abrogate it, because you cannot abrogate metabolism any better than you can abrogate aging. But you can get these animals to live long, and more importantly, in those animals, not one, but many signs of delayed aging will be present. So they will be having increased motility. They will be showing better flexibility of their tendons, of their joints. They will have no arthritis. They will not have cardiovascular disease, or at least all of that will be delayed by many, many months in their lives, which will translate to a couple of decades, if not more, on our lifespan. And so all of these are connected in that way, that they plug into the chronic disease. And then the idea is here, rather than to shoot one bullet for every target. Our target is to lengthen not the lifespan per se, but really health span. Those years that we can live healthy. And that magic of all of these animal models showing, including non-human primates, and even in some studies, some signs that this might be happening in human, are allowing those models to live not only longer, but live healthier. And that is, of course, the goal. The goal is to live longer lives, not indefinitely, but whatever we have, to live with maximum function, and then to have hopefully only a short period of decline, before we have to say goodbye to this world. And a lot of evidence is showing that this indeed is possible. So there's this concept of health span that we are operating with. And so here, if you were to review medical charts, if you were a doc, and if you looked at these two people just based on the charts, you can find very similar things. Both are the same age. Both have hyperlipidemia. They had prior myocardial infarction, so their fats are elevated and all of that and stuff like that. But if you look at them, there's no doubt that one of them is a lot better off than the other. And that is that concept of resilience and of heightened function. And so the goal, in a way, here is, and this is based on what we are fairly confident can be achieved, that we can have a person that looks fairly similar towards the end of life for a very long time. Now, we are now at the point where at least about 5-6 years ago, the expected lifespan for a female in the US who's now 50 is about 81. That is continuously lengthening by some amount, but that's not followed necessarily by the lengthening of the health span. And in fact, there's some cases where the length of life progresses, but the health span actually shortens. And that's not what we want at all. We really want to get to this point where health span, and this number of 114 years of age is based both on supercentenarians of people that live the longest, where can they actually go, where can they get, and also an extrapolation from many animal models where interventions have been performed to increase health span and lifespan simultaneously. So whether that's exactly true, whether this is there, or whether it's like 105 or maybe 120, it's kind of like a matter of experimental error, if you want to call it that way, but it's definitely in that ballpark. The most important part would be that person would be able to function quite well. So let's now turn, and there's a lot of exciting stuff. We can, set up another meeting where I can tell you a lot more about that and what's now on the cutting edge, and what, there are interventions to eliminate old cells from our bodies. One theory of aging says that we grow old because we accumulate old cells. Now, those old cells have been very, very difficult to find, to the point where that theory was not considered even viable. Now we know that there are some cells that look old and that you may be able to eliminate through the process and the interventions that are called senolytics. Whether senolytics will be able to extend our natural lifespan and health span, we don't know. But they seem to be efficient in clinical trials at getting at some more acute problems, like, in folks that have issues with the chronic obstructive pulmonary disease or idiopathic pulmonary fibrosis condition that doesn't have any cures right now. This seems to be actually reasonable and efficient. There are all sorts of diet modifying interventions, whether it is, controlled fasting of some sort, you eat for just a period of your day, or you eat just for a period of your week, and there's a part of it where you don't eat very much, you reduce your calories and all of that. And whether you use modified calories or modified diet in terms of different mimetics for the diet, all of that is very exciting. All of that is gaining more and more clinical support. So at this point, I will turn my attention, and yours as well, to our immune system, because that is something that we study directly in the lab. We do study some of these health extension modalities, but when we study them also in the context of the immune system and what does it do to the immune system. And so immunity and inflammation have been recognized, inflammation in particular, as one of these basic pillars of aging. And if we can control our inflammation well, there's also a very good chance that we will live longer lives and healthier lives. So the immune system is, unlike most of our other organs and systems, a distributed system. It runs and basically services every part of the body. And its components are traveling and trafficking at high speeds so that they can be at the right place at the right time. Some of them sit for a while in certain places and then they say, well, it's kind of boring around here. Let me check what's going on elsewhere. And then they go to the other parts of the body. But the idea is to have this patrolling function of the immune system. And so here you can see in blue a whole bunch of little lymph nodes. Lymph nodes are the units of immunity. They're sort of like the garrison posts, if you want to call them that way, where immune cells congregate. Many cells sit there for a long time waiting for the infection to happen. That's also where they have to be maintained, which is an important point. Long-term maintenance is, of course, a key part of successful aging. But also when the infection finally hits, that infection will be focused into the nearest lymph node relative to the site where the invasion has occurred. So if the virus is coming through the nose, there is nasal lymphoid tissue right around the nose where this initial reaction will start. It goes deeper into the lung. There are lymph nodes that drain the lungs. You know about the skin draining lymph nodes. You can actually feel them, or you could have felt them. You know, all of us could have felt them when we were younger, you know, starting with the tonsils or the ones that are on the neck, or if you get cut, you know, then maybe behind your knee or lymph node will swell and so forth. So lymph nodes, spleen are the organs that actually are in charge of maintaining the lymphocytes and starting the reaction. But lymphocytes themselves are made in two other sites, in the bone marrow and in the thymus. And the bone marrow is the source of all cells, except that some of them stay there and make B lymphocytes, which will make antibodies down the line, whereas some cells migrate to the thymus, which is right above your heart, this yellow little thing here. And that is the one that's going to make all of your T cells. And B and T cells are really important parts of our adaptive immune system. So let's talk a little bit about this. So the challenge for the immune system is to always be at the right place at the right time and to outrun the infection when it starts. And to do so, we have this system of meeting spots, the lymph nodes and the spleen, where immune cells will start the immune response. These are the cells of the immune system, so don't get intimidated by any of this. We're going to go through this one by one. The important scale here is the time scale at the bottom. It's hours to days, and normally there's no infection in our body. And at that point, all of our barriers are intact, our skin is intact, our mucosa has not been invaded, and so forth. But when the infection strikes, then this little thing here, and let's say that this is a virus, will actually enter. It will cross the body barrier. Whether that barrier is in your gut, whether it's in your lung mucosa, or whether you get cut and it enters through the skin, or whether a mosquito pricks you and actually infects you with a virus that it's carrying or something like that, that is the breach of the barrier. And then at that point, there's going to be a whole slew of cells that live right underneath these barriers that are going to be in charge of immediate alarm. They will be the ones that initiate the immune reaction. And those are the cells of our innate immune system. Innate immunity is something that the name says we're born with, and it doesn't get too much better with re-exposure to infection. Unlike the adaptive immunity, which can be trained to get better, better, and better. It's kind of like your muscle. You know, if you exercise it, it's going to get better. Same thing is true for adaptive, but not for innate immunity. But innate immunity has this fantastic ability to react momentarily. It's literally going to be seconds to minutes where the first recognition of infection is going to occur. And so some of these cells will be gobbling up this virus to remove it. Others will be calling for alarm by producing different types of signaling molecules, recruitment molecules. And all of this is happening minutes to hours on this scale. there are molecules also that are going to be involved that are floating in the extracellular fluids and so forth. But this one cell here is super special. Where did I lose it now? Okay. Yeah, sorry. So this cell is super special because this is the singular cell called the dendritic cell that can actually eat up the pieces of dying tissues and of the virus in this case, or bacterium, whatever has attacked us, and bring it to the lymph node where it's going to be presenting it to the T cells. And then the free virus will be floating there as well, and it will be activating the B cells. And the T and B cells, once they're activated, and they're going to need much more time, are going to really allow us to eliminate this infection. Sometimes the innate immune system can do the job by itself, but in most cases, that's not going to be true. And in most cases, we will need this adaptive immunity. And it's interesting when you think about how this all came to be, that this immune system actually arose when vertebrates evolved. So when critters that are going to be not stationary, but highly, highly mobile and venturing all over the world, essentially, which is what we do to our peril very often, we will have that extra layer of immunity that will be protecting us from that, which we're now a lot more likely to encounter. And so the consequences of this activation, and interestingly, these two cell types will also collaborate at some point, is that B cells will make antibodies. And antibodies are fantastic molecules. They can bind to this virus and they can whisk it away, or it can make it more appetizing to be eaten by phagocytic cells like these guys here, like macrophages and neutrophils and so forth. These cells are the key orchestrators of the immune system in many ways, because they will both directly attack the infected cell, and they can kill the cell, because if the virus infects the cell, the virus cannot multiply outside of the cell. It has to actually usurp your cells to make more of itself. It turns each of your cells into a virus-producing factory. So what the T cells do, the ones that are so-called the killer cells, they will figure out, okay, this cell is infected with the virus. If I let it go for another 12 hours, it's going to spew out another 100,000 copies of that same virus. So I'm better off killing this cell for the time being, and then we will regenerate that cell if we can down the line. These cells will also give help to these B cells to make better antibodies, and they will do all sorts of other things down the line. But we'll talk about that. But they need days because we have very few of these cells at the beginning of infection. You cannot have a ton of these cells specific for every infection because our body is finite. We only have roughly about, I would say, on the order of about maybe 10,000, maybe 20,000 cells specific for any particular bug. We need millions to fight them efficiently. So during this period here, there's going to be crazy proliferation. Like these cells have to divide, make up more of the cells like themselves. They also get armed because in this state here, when we start the whole story, they're so-called naive cells. They haven't seen their bug, and they're not really able to do anything about it. But by the time about 3-4 days of proliferating and differentiating, they're now to become very angry, and they're going to be making a lot of antiviral molecules, and they'll be able to do a lot about it. So again, we can skip this. I think this one got here for the wrong reasons. It's just a reminder to tell you more about the lymph nodes. So most of this second phase is happening in the lymph nodes, as I said. So the dendritic cell comes into the lymph node carrying the bug, and it's chopping it up and it's displaying it on its own surface so that T cells can meet it. And that's happening here in the middle of the lymph node. B cells are in another part of the lymph node, and they will meet later. I'll show you what's happening here very soon. And about 2% of the lymphocytes always move between the lymph nodes. Again, because if one lymph node, like, let's say that you got cut on your big toe, it's going to all happen behind your knees. That's the nearest lymph node that we have, the popliteal lymph node that's right here. Obviously, the ones that you have in the pinky are not going to be affected, right? Or in the lymph node around there. So they're going to be called, and they're going to migrate, and they're going to stick in this one big inflamed lymph node because that's where the action is. There's going to be a process of the call to arms of all of the cells to recirculate and get to the site where we need to maximize that defensive reaction. So what are some of the problems that we're facing when we're older? clinicians will tell you immediately that there's a lot more people that get sick or that die from infection, particularly if it is the infection that we have encountered for the first time, that we do not have pre-existing immunity about. I told you that you can train adaptive immunity. What is even better about adaptive immunity is that you can actually make it remember, if you want to call it so. That is called immunological memory. And what that means is that after you have eliminated the immediate threat, there's going to be a large-ish cohort of cells that survives for very long periods of time. And so should you get the same bug very soon, those cells are going to protect you. And that is the basis of vaccination. We're faking the first infection by giving the vaccine. We're giving it in an innocuous form. And what's happening then, you're mobilizing all of these cells, you're making them all differentiate, and then you're going to keep some of them for very long periods of time in your immune memory. That's what provides protection against the real infection that comes after that. That's how the vaccines really work. So what's happening here is that, if we haven't had that benefit of vaccination and we have not met this particular bug when we were younger, now we're facing it for the first time when we're older. And our ability to respond very robustly is eroded. And we'll talk a lot about how and why exactly. But the levels to which this is happening, I probably don't need to talk to you about it, but I will give you the exact numbers, are staggering. Because this high susceptibility to emerging in new microbial pathogens is increased relative to the younger population, somewhere between 10 and up all the way up to 200-fold. And so the numbers here are for SARS coronavirus 2, if you're not vaccinated and if you are above 80, your chance of dying from that infection relative to the population between 18 and 39 is 270-fold. It's not 270%, it's 270-fold. And that's enormous. And the same is true for West Nile virus when it swept through the country, and we were all naive immunologically to it. It was somewhere between 43 times and 50 times that you would be more likely to die of encephalitis, which is what this virus does. It goes to the brain, causes inflammation of the brain, and unfortunately, some people die from it. And so that is one problem. The other problem is that for very same reasons, our response to vaccination is not all that great. Now, the good news is that this is not a doomsday scenario, because we are now learning a lot more about what to do with vaccines and how to make vaccines that would be better and a lot more efficacious in the older population. And these SARS coronavirus 2 vaccines are amongst them, although we do need to study them more, particularly in terms of longevity. They have been shown to produce very robust immunity in most older adults as well, if there's no additional immune defect. So what is happening here, again, the best and most prominent are the changes in adaptive immunity. Sorry, I missed this one. So many components of the immune system are slightly less quick, slightly less numerous, and slightly less robust when we grow older. The problem is that this is a reaction that has to happen in a precise sequence. So if step 1 is like 5% diminished, step 2 by another 10%, and step 3 by maybe 12% or so, this is a cumulative problem. So at the end, all of them will add up to cause something that might be 2-fold or 3-fold worse, relatively speaking. So again, the innate immune system does go down in robustness, and we're still understanding that because that research started relatively later. But there are problems in sensing of the bug, signaling, and effector functions, elimination of the infection. We understand better the changes in adaptive immunity that are in charge of eliminating the infection in a highly specific manner. So again, one thing that I didn't tell you is that the innate immune system just wants to know, am I dealing with a virus? Am I dealing with a bacterium or with a fungus or something else? And that's all of its specificity. It's not a highly specific system. So it acts very well to contain initially the infection, to alert the adaptive immune system with what's happening. Whereas the T and B cells are laser specific for that particular virus. So what you mounted against SARS coronavirus 2 is not going to be specific for flu. You're not going to get any reactivity against smallpox or any other bug. It's going to be really based on very highly specific receptors that recognize pieces of the virus. And so we already covered this, that new infections not encountered before will require the response by naive, previously unstimulated T and B cells. And that this is actually where our main problem really occurs with aging. The responses that were generated earlier in youth that we have encountered when we were younger are dealt with memory T and B cells. And those cells are maintained fairly well. And in many cases, you guys might remember that there was quite a bit of panic, but fortunately it never turned out to be as bad in 2009 with H1n1 bird flu at that time. For that bird flu, if you look at the curves of mortality, who died and who didn't die and what happened, we had an inverted curve. We had older adults do really, really well because they have seen that strain before in the 50s and in the 60s. Whereas it was really the segment of the population after them that was immunologically more naive, and there was relatively more death. That was an example where we had pre-existing memory that kept us, well and healthy. So the naive T cells, their naive lymphocytes in general, decline in numbers in the last third of life for two reasons, and we'll talk about why. And they leave us potentially vulnerable, and we'll talk about why and how this vulnerability occurs. So the T cells, which I told you are really important as coordinators of the response, are prominently and reproducibly affected by aging. So we can talk a little bit about what's wrong with old naive T cells, and is something wrong with them or with something else, which is really important when you try to think about how to fix the problem. You really need to know, if you're a plumber, you need to know, is it like, the little valve that is, being eaten up, or whether the pipe is rotting or what's going on. It's similar principle here. We want to try to design something, we need to know what's going on. So this is in a way what's happening really in the adult. This detection of the virus, and again, the virus are these wonky little things drawn up in the corner, is happening by dendritic cells. And I told you that they pick it up, they eat it up, and then the moment that they eat it up, they become pretty angry themselves. They get bigger and they move from the tissue to the lymph node. When they get to the lymph node, they will meet quite a bit of naive T cells. And these naive T cells will proliferate a lot once these two cells meet, and they will generate this large pool of cells. And you see these little tiny granules here. That's to depict that they are now really armed and dangerous and that they can deal with the virus. Now, what happens with aging is that this sensing, first of all, is diminished. So that the dendritic cell, I'm sorry that you can't see this. You may want to. Oh, good. That works better. This leads to a weaker maturation of the dendritic cells, impaired movement. So they don't quite get as fast to the lymph node, and they're not quite as activated. But even more of an issue is that this pool of T cells that they meet there is already diminished. And it is diminished because the production of naive T cells already by the time of puberty goes down by tenfold. We've got only 10% being made after puberty. And it's, maybe okay if we can maintain them for long periods of time. But there is also another drop that occurs somewhere between the ages of 40 and 50 that knocks that down, but overall from the beginning to only about 1%. So we didn't lose the thymus, we didn't lose its ability to make any cells, but it's not making nearly as many as it used to. And so now you have to maintain these cells for many, many decades. Whatever you got, by the time you were getting into your early teens, you want to carry it for another 70 plus years or more. This maintenance is working pretty well until the last third of life. And then there are signs that there's faltering of this maintenance as well. So all of these things are important. So if the naive T cells are lost, how about we make more of them? Can we do something like that? Could we reawaken the thymus? That would sound like a pretty straightforward thing to do. Let's give it a shot. And there are, it has been known that for 70 years that castration can actually transiently regrow the thymus. Now, it doesn't sound like a great perspective for treatment, but in the meantime, of course, we were a little smarter than that, and there are chemicals that you can take temporarily that will have the same effect. They will block sex steroids. And for example, many of them are FDA approved, like, for the treatment of prostate cancer, for example. There are some anti-androgen treatments. And so we could try to use this, and we can actually, that will allow us to transiently increase the thymus size, cellularity, and new T cell production. And, that lasts for about 2 months in mice. But if you do extrapolations, that would be equivalent to about 5 years in humans. And I would be perfectly happy to have 5 years of new T cell production. It will last me probably for a while. So the question is this enough to improve immune defense? Because really what we're looking for in the end is better defense against microbial infection. So this is an old thymus in the mouse, and this is the adult thymus. You can see what a difference that is. It's pretty huge. You really lost about 10 times or more of the mass. And when you look inside of the structure, it's even worse, because this one's like chock full of fat and fibrous tissue and so forth. And so we've used this substance, Degarelix, which is one of these drugs that's actually used for prostate cancer. And when you give Degarelix to old mice, you can see how much we can regrow this organ. It's roughly comparable by weight and a lot of other parameters to this adult untreated thymus. Now, if you treat the adult thymus, you can make it even bigger, but that's not really the point of what we're trying to do. So then when we looked at what's happening in the thymus, this is the number of cells in the adult thymus, and this is the number of cells in the old thymus, at least the ones that we care about here, the ones that are about to go out to the lymph nodes to rejuvenate and repopulate the naive pool, as we call it. And you can see here that when we treat with Degarelix, all of this comes back up. And the black and the white are just for the two flavors of T cells, CD4s and CD8s, that do slightly different things. And when we wanted to look at these same cells in the blood, which is how they're supposed to traffic, and then also in the lymph nodes, which is where they're supposed to live and function, we were a bit disappointed, because we saw sometimes significant, sometimes not sign that they were in the blood. But when we really looked whether they were in the spleen and the lymph nodes, we didn't see anything. Again, this is the adult, this is the old Degarelix treated. Now, when we label these cells, we used some tricks to label them. We could see that some of them have made it there, but the total numbers didn't improve. And we weren't sure that we have done a whole lot for this particular mouse. And I don't really trust any of these results unless we have an infectious model. And we have some years back actually generated and characterized mouse model of the West Nile virus infection, which is very similar to what happens in older humans. And so when we do that, we know that adults survive roughly at about 70 to 80%, and that old survive only at about 30%. And unfortunately, when we gave these mice Degarelix, so this is with the old without Degarelix, this is old with Degarelix. If anything, it was probably a little worse. It wasn't quite significant, sometimes it was really worse, but definitely wasn't better. And that really made us worry about what happened here. Why are these cells being made by the thymus, and you can find them in the blood, but they're not really moving to the lymph nodes? And so then the question, of course, is what's wrong with the old lymph nodes? Is there anything wrong? So you can just line them up. This is the spleen, this is the thymus, this is the bones, and these are the lymph nodes. These are the adult ones here. You can see that the old ones are quite a bit smaller. And if you talk to the geriatricians, and we all know that actually from experience, that lymph node swelling really doesn't happen to us very much after we turn 50 or so. In particular, if we get older, you get infection, you're not going to get a swollen, painful lymph node as a rule. And so that also told us that we might actually be onto something important to look into the lymph nodes. So the lymph nodes, I showed you that structure. In addition to lymphocytes, lymph nodes have a whole slew of really important cells that are there to service the lymphocytes, to provide the cushy, nice environment, give them, good nutrients, good trophic factors to keep them happy, and also to direct them against each other. It's quite fascinating when you look at it. It looks like a superhighway. It looks like a 3D mesh where the cells are, these stromal cells that make this mesh, elongate towards one another and connect, and they make like this, beautiful, 3D structure. And then the T cells are crawling around picking up nice little nutrients. When the dendritic cell comes with pieces of the bug, it will crawl towards the T cell, which allows them to meet in a directed fashion. You can imagine if they were left without any support, they'd be floating in space and they would never meet each other. Whereas in this case, actually now they are, having a much higher chance of meeting one another. And this is the staining of this meshwork structure. I'll show you some more so that you can see. But when these cells are actually pulled out of the lymph node, you can dissociate it, you can digest it, and you put them together with T cells. The T cells live a lot longer with these stromal cells than without them. They die off really pretty quickly unless you give them these stromal cells, as we call them. And that's the case for two major subsets of T cells. So then we have done this experiment in my lab. This was an experiment from another lab. So we knew that these stromal cells were important. We were really shocked when we started looking for these cells. So at 3 months, these are the lymph nodes, and we can stain these regions with different molecules. We can look where the T cells are, and they're violet. We can look for in red. These are the stromal cells. And when you magnify these little squares here, you see that at 3 months, you have this beautiful connected meshwork in different lymph nodes. It's clearly there. These are the skin lymph nodes, 9 months. This is equivalent to probably about a 35-year-old human or something. And this meshwork is completely gone in the skin draining lymph nodes. In the ones that drain deeper tissues, it's still very much there. Now, this mouse here at 19 months is drawing Social Security. It's bona fide old. So in that one, now you can see that even the deeper ones have this deterioration in structure. And so that for the first time started telling us that the supporting structure that the T cells need to be maintained for a long time and to actually do their function, to do a good recognition and reaction to the microbial infection is really deteriorated. And so there are parallel studies by other groups that have shown also that the T cells, when they get into the lymph node that's like that, move very slowly and are quite disoriented. So it takes them a lot longer to find their dendritic cell and to do what they're supposed to do. So now we're actually really interested in trying to fix this through different ways and means. So it's not only the thymus that we need to fix the lymph nodes. And so these are our conclusions. We lose many naive T and B cells with aging. This is a problem with mere numbers of lymphocytes that we need to respond to infection. But the problem is also in their coordination and maintenance. Even the ones that are around, which is only about 1/3 to 1/5 of what we had in youth, do not work well because their maintenance, guiding, and trafficking system that exists in the lymph nodes deteriorates with aging. And so we really need to fix both. And so the good news about all of this is that aging is a plastic process. It's not something that happens once and you can't do anything about it. There might be optimal windows for intervention, and that for some of them, we might need to intervene at middle age rather than when we're already old. But we know for the immune system is that if we provide some additional juices to the T cells and in the lymph nodes and so forth, that they can quite magically make all of this work a lot better. Because there are additional defects that occur in the process of recognition. Some of these cytokines are not being made. Cytokines are the hormones of the immune system. And we now understand that some of the vaccines, not all of them, but some of the vaccines, and we've known that for a while, but now we're testing it actually experimentally, both the SARS coronavirus vaccines and the new varicella zoster vaccine, the Shingrix vaccine, are very good at actually generating good immune responses in older adults. And the reason why they're being able to do that is because they're stimulating very good initial innate immune response that then provides all of the juices to the T and B cells to be able to react a lot better. Now, the side effect of that is that none of us feel so great when we get those shots. You're feeling like you're knocked out for a little while, because it is that reaction that you're feeling is really the reaction of your innate immune system. It always has been, even whether it's the infection or whether it's the vaccine. And so good, strong live vaccines that we have used for a long time, like yellow fever and like smallpox, will give you that reaction anyways. Except that, they were old platforms, so they're really actually quite a bit dangerous themselves. I mean, it's always like, if you get a disease that's killing 40% of the population, you're going to take that vaccine and not worry about it. But if somebody's like a little bit immunosuppressed, then they could have problems with those types of vaccines. We're way past that, and our engineering of vaccines now has, it's paradoxical that more and more people are hesitant or hostile to the vaccines, and we're making better vaccines than ever and safer vaccines than ever. We'll talk about that later. So anyway, the long and the short of it is that there's a lot of ways in which we can intervene. That will be the story for another time in greater detail. Let's talk a little bit about this virus that we have been dealing with for a long time now. You all know what symptoms it gives you, and I will be taking you back in time to late February, early March of 2020, where we have decided that we want to study this virus. Obviously, my lab is studying immunity in older adults, and when there is a virus that is so difficult for older adults, we have to jump and start studying it. So can we detect good immunity to it? Can we figure out who was exposed, who wasn't? At that time, the tests that were on the market to measure antibodies were horrible, and the incidence of the disease was very low. So that provides, I'll tell you why that is a particular problem. But we had to make our own test in the end, the long and the short, and I'll tell you about that test in a second. We wanted to know what is the duration of natural and vaccinal immunity, and is there an immunological base to the vulnerability of older adults to this virus. So in making the test, my laboratory teamed up with Deepta Bhattacharya, who's a professor in my department, and Deepta's a B-cell immunologist. My lab traditionally studies more T cells, but we had the expertise between the two of us to get on that quickly. And then we teamed up with Ryan Sprisler, who's running a clinical lab at the university that was helping us do something else. And as we started developing the test, and I don't know who got the first whiff that we're doing that, but there was a reporter from Arizona Republic that interviewed Deepta and myself, and I have it in the print, what I have said at that time. And it says here clear that UA antibody testing will not be used to conduct widespread testing in the population to figure out who has and who hasn't had the virus already. It will be used for research, and it will be for coordinating with healthcare providers. So that was 28th of March in 2020. And this was April 14th, where we had an oh crap moment. And we had no idea at that point, how are we going to do this? Because this was the time when we were validating our test to make sure that it's really telling us positive from negative. We were adding additional controls. We had, testing this number of people was beyond our wildest dreams. And thank God that we had Ryan Sprisler with his genetics core, which is a beautiful lab that has a lot of these robots. And we could retool the robots rather than to do genetic testing, that they could do antibody testing. And it's really a fun, if you come to campus, we can show you how this looks like. And we were able to scale this up to do 3,000 tests per day, essentially. And as a consequence of it, we're on the tail end of that contract with the state. We have tested more than 60,000 people. Didn't quite come to that 250,000, unfortunately. So what was the test, and what did we do with the test? I mean, the advantage for us was that, unlike pharmaceutical firms that wanted to put some product there and make the money, we had the advantage of actually critically looking at whether our test is really working or not. And so everybody was targeting their test to the most variable part of the spike protein, the so-called receptor binding domain, or RBD. This is the part that the virus uses to latch onto our cells. It binds to this ACE2 molecule, which is expressed on our lung cells, nose cells, and everywhere else in the body, actually. But that wasn't enough, because, when we did this assay and when we started looking, these are the RBD antibodies here. We couldn't quite figure out where is the cutoff here and who's positive and who's negative. And we could get to about 98% reliability in terms of who's positive. But at that point, only 1% of the people were infected. And so that means that you're really in the crapshoot zone. You have 50% chance of telling somebody wrongly that they're not infected, or even worse, that they were infected, and then they would assume that they would be immune and they will be going out licking doorknobs and stuff around. So what we had to do at that point, we figured out that if we introduced another test that would measure antibodies against a different part of the virus that is distinct from RBD, then we could get just the people that are positive by 2 criteria, not just by 1. And what we show here is that this is the binding. So everybody in this box was above our 2 cutoff limits. So if you were positive for RBD, we would test you for the S2 part of the spike protein. And then if that was positive, you would fall here and we would say, yes, you are positive, you had an exposure. This was way before the vaccines were around. But the most important piece for our peace of mind was that we did this other test that allowed us to color these guys red. This is the antibody neutralization assay. And what that does is you basically take the virus and you put it together with the cells that the virus would normally bind to, and it would kill them all. It would basically pulverize them. So you would see, there will be a violet monolayer of cells and you would see these pocks in there. That's why we call it the plaque assay. If you put an antibody that blocks the virus, that neutralizes the virus, the cells will stay alive and there will be no pocks forming on that monolayer. That is what we call neutralization. The antibody has neutralized the virus. It has completely blocked it from even entering the cells. Those neutralizing antibodies are the ones that we want to have. And those are the ones that vaccine manufacturers are trying to generate. And so the super comforting for us was that everybody who scored, whoops, that was bad. So runaway slide, okay, fine. Everybody who scored above these 2 limits also had neutralizing antibodies. And that made us confident that we really have a good test. To this day, this is the only test of this type that has been made and is one of the most accurate ones in the world. So we use that and these are some other details of the testing and what it was and so forth. The interesting point was when we then used this test to try to understand the limits of immunity and the durability. When the B cell gets activated from the naive state, it has 3 fates. It can generate this short-lived plasma cell. This is the SOS wave of antibodies. You are fighting for your life with an infection. You want to get some antibodies out there to fight the virus as soon as you can. These are not the best antibodies that you make. They're not the most long-lived antibodies that you make either, but they are the ones that you can throw out there quickly because B cells are the slowest part of the adaptive immunity. T cells actually get in on this first. Some of the B cells will go and actually do a lot more after this initial stimulation, and they will give rise to this memory B cell and to the long-lived plasma cells. So there's a second wave of much better antibodies and much longer-lived antibodies coming after the first wave. Unfortunately, many of our colleagues who were publishing and doing work and in the field, and many of the journalists really didn't ever learn this basic immunology, because this has been published for a long time. This tells you that this is the half-life in years for different infections and different vaccines, how long your antibodies live. So this is the so-called half-life. So when you get a tetanus shot, you will lose about half of the antibodies over about 10 years, which is the point where they make you have another shot. And if you go down the line for diphtheria, that can be a little longer. Some of these are remarkably long. So we know that for measles, after the vaccine or natural exposition, we actually cannot detect the loss at all. So you would lose about half if you're a male in about 370 years. Now you've got quite enough. And females don't seem to lose any, but we knew that they were better. And so goes for all of this. So thinking that a dangerous infection like SARS coronavirus 2 will not give you long-lasting immunity really ran against everything that we knew about immunology at that point. And nonetheless, there were titles like these in lay press derived from the papers that were published in scientific journals that really didn't tell they had the data. Their data weren't wrong. Their interpretation was completely wrong. They were saying that we'll lose in 3 months all of the antibodies that we have. Now, why did they do that? Because this is an important point here. This is not Bob Dylan, and he wasn't a drummer. He did not play for Metallica. So, and you will find things that are very similar to this type of information all over the social media. When you measure the initial antibody response against SARS coronavirus 2, you get this peak that goes up. This is just the concentration of antibodies that you can measure. And we know that this extrafollicular response, the short-lived response, will last for some part of this period. At this point, in every infection, in every vaccination, these amounts of antibodies will go down. And that is what they measured. So their measurements were not wrong. What they didn't do is they didn't have this part of the curve to measure, and they extrapolated from this drop that this is going to crash outright, completely ignoring the fact that there is this germinal center response that generates antibodies that are 100 to 1,000-fold better at binding the virus and are much, much longer lived. Now, we don't know exactly how long lived, but they would be definitely here in the realm of years. So that was, one of the problems. And so then we did our study using our test to look not only at people that came out of the ICU or that had severe SARS, but that had very moderate or mild or even asymptomatic that we could find were positive. And then we looked at how much of the antibodies they had over about 6 to 7 months. And you indeed see this peak, and then it goes down, but it's going down very slowly, whether you measure it against the RBD or S2, or most importantly, if you measure the neutralizing titer. The neutralizing titers were remarkably stable over that period of time. Now, again, how long exactly this will last depends on a lot of factors, and we don't understand everything here, but I'll share with you what we do understand about both the vaccines and the natural exposure. So what we know about antibody durability is if we look at the people who got the Wuhan strain early on, they keep their neutralizing antibodies against that Wuhan strain all the way to now easily, and they don't lose much of the titer. So if Wuhan strain was to come back again, there's a great chance that most of them would never feel it. The problem, of course, are the variants. The virus didn't decide to go into retirement. It did what all viruses usually do, which is try to mutate. Depends on what type of the virus, but RNA viruses like this one that are relatively small are very good at mutating relatively quickly. This is not the worst one in terms of the speed of mutation, and it actually even has an enzyme that's kind of like constraining its ability to mutate wildly. And that is because if it mutates too widely, it will lose a lot of capacity to do other things that it cares about, most notably to infect a lot of people. If you think about what the virus tries to do, it's trying to be super efficient at infecting as many people as it can. And in the process, it's going to be better and more fit from its own standpoint if those people don't die right away. So the evolution here, we had the case of this virus jumping from several non-human hosts, going back and forth between the bats and whatever other dogs and whoever else was in that Wuhan market, and getting into humans that were completely unused to it. But guess what? The virus also wasn't used to us. So what you have seen through viral evolution is that it increased. It was, first of all, when it started, it was a bit better than regular flu. Actually, it was a little better than pandemic flu of 1918 in terms of spreading between people. And then it went to the Delta variant, which was really a lot better. Delta variant was overall ended up being about 3 times better than the original Wuhan strain. Measles are the legend amongst the viruses in terms of how well it transmits. Measles transmit with the reproduction factor of about 15, which means that one person with measles will infect 15 people around it. And because the measles are distributed in these clouds, if somebody coughs or whatever, you can literally walk into somebody's cloud walking on the street and really pick it up that way several hours after that person hasn't been around. Or walk in the room, probably more than outdoors. But those are very important considerations when you think about what's happening. Now, Omicron doubled what Delta did. Omicron got to about 11, her reproduction factor of 11, which means that it actually really has approached measles. And I thought I've seen kind of like everything. We all think that we're somewhat smart, and we bet on how some things are going to happen. I did not really think that we would face Omicron, but then Omicron did something that, again, as I told you, was not too unexpected. It lowered its pathogenicity. It was selected because it was transmitting well, and it wasn't really killing too many people. Again, both things are favoring virus spread, which is what the virus is trying to do. So we next wanted to see what's happening with our mRNA vaccines. How good are they, particularly in older adults? So we did this study. Now finally it was accepted for publication, and some of these things took really forever. I mean, were many abnormalities, and including the scientific publishing. Some things went into print very quickly, some went ridiculously slowly. This one we submitted back last June, and we just learned of its acceptance and whatever. But an important point is that we looked at first 2 doses. One dose was here, one was up there. And the broken lines are older adults, people over 65. And the younger group here was relatively, it was under 55, but really the median was around 38 or so. And you can see that when we looked at the efficacy of the vaccine against Washington, which is the nearest cousin to Wuhan, it was really beautiful and no difference in their reactivity at all. When we looked at the response against Gamma, this was the first point where we saw significant loss of antibody binding. And this is neutralization. So this is the one that we consider the most relevant one. So relative to the younger population, there was a significant loss of binding here. We used another strain. We repeated it with Delta down the line. All of this showed that there was some loss with antibody binding. Good news was that the T cells were really, really resilient against these mutations. So you can see that regardless of the age and regardless of the variant, and this is Brazil and this is Delta here, the B1617, relative to the Wuhan original strain. If anything, older adults were, maybe even a little better by a smidgen. So T cells are the ones that are going to keep you from dying, and they're the ones that are going to keep you from severe disease. They're not going to be able to whisk away the virus so that you don't get the infection. And that goes along with the fact that the main target of antibodies is the spike protein. This is what's in the vaccine. And the main thing that the virus has been changing, because it has been feeling this pressure, has been also the spike protein. And that's how you evade antibody immunity. You remember, you have to have those antibodies right up here. This is the entry point, nose and throat. It's not like SARS-1, which was like deep in the lungs, which is the reason why SARS-1 never spread beyond the 8,000 people that it infected. It was a whole lot more deadly, but it didn't really spread very well, whereas SARS-2 up front was super awesome in terms of the right site from which you can infect, and also the site where it's not that easy to have high concentration of neutralizing antibodies. In order to get to that, and I'm talking about that down the line. So we've also studied some of the breakthrough cases because we wanted to know what's happening. This dovetails with this same story that I'm telling you. We looked at the neutralizing titers in controls and in the cases, and you look at the parental strains. So this is the Washington, and it's a little lower in the cases, but not by much. So these are the numbers of people. There's a ton of people piled up here that you cannot see. These are all supposed to be original dots. So you've got probably about 20 people here, about 15 people here, about 3 folks here, and so forth. So if you compare this to this, it's not much lower in the breakthrough relative to here. If you look at the Delta, because these were breakthroughs with the Delta, they are lower, but you still have 3 people that have a titer that's 1 to 80, which means 180 times more than what you would need to neutralize in the Petri dish. Now, obviously, we're not Petri dishes, so that doesn't quite exactly translate. But in others, infections and in other vaccines, if we had that titer, we would have all bet that everybody's protected. at 1 to 80, but not with this one. Now, why is that exactly the case? There's several reasons for that. One is that this virus has evolved really, really well to bind our cells. The strength with which it binds to the receptor on our cells is legendary. It has been 20 times better already in the original SARS Wuhan than it was in SARS-1. They use the same molecule to get inside the cell. And then every mutation that has lived in the population, starting from alpha, from the so-called UK strain, has gotten better and better at getting into the cells. So to be able to really block the entry into the cells, you have to have super good antibodies. And you have to have a lot of them. And again, the problem, as I said, is you have to have them up here, not in the bloodstream where everybody has them, actually. So we need to probably formulate mucosal vaccines for that. Like, remember for polio, you get little sugar cube and you eat it. Yeah. That actually makes antibodies in your GI. That is where your immunity against polio is. And that is why that vaccine is so good at making sure that you don't get infected with polio, which is an enteric virus, which gets into your gut to infect. Now, this is actually a little more different. You need something to sniff probably or something like that we'll need to do. But that's the point. That's the important point. So reinfections may be the cases of response breadth also. We don't know how broad are these responses. That's one of the things that we're really trying to study. We have a study at several senior living facilities where we're trying to make sure that we have serial blood draws from people, try to catch them at second vaccine, third vaccine dose, and then anybody goes into the breakthrough. And we're going to, because we specifically want to study that breadth of the response, if a new mutation occurs, how well can you contend with it? Because there's been a lot of talk in the literature and a lot of speculations, and I'm guilty as charged about speculating about that it is the breadth of the repertoire that also narrows down. We know that it does as we grow older, but we don't know how much, and we don't know how biologically important that is. So that's something that we need to study for sure. And then magnitude and durability. So breakthroughs are very rare against the original infecting variant after natural infection. But since the virus is evolving, that is where our problem is. And again, the antibody amount at the site of infection is important. And then even if the vaccines are not preventing all of the infections with Delta and Omicron and so forth, they remain very efficacious against the disease and reduce the probability of severe infection and death very well. And so, again, there's a question, why are we not more vulnerable if you look at the antibody data? And I told you that is because T cells are very resistant to virus mutations. So I showed you the data up to Delta, but we actually have just gotten another paper accepted on the Omicron with relatively a large cohort. So Omicron, after 3 shots of the vaccine, was very efficiently prevented by T cells and recognized by T cells. And so again, there's questions, what does this mean relative to the end of pandemic, to the future living of this virus, to the numbers of shots that we might need down the line, disease duration and so forth. All of that is, has to do with how we live, with pandemics. So let's talk a little bit about that. So our vaccines are good at doing any of these. So they're preventing hospitalization and death in most people and symptomatic lower respiratory infection. Those are the ones that are really going to make you sick. Now, you want to have enough shots, of course, because there is this nasty little thing called long COVID, and there are other things that can mess you up with this. We do have a big grant that we have received to study long COVID. Those studies are in process. We are recruiting. I am, guess what, the principal investigator on that one as well. But it's a very important program, so hopefully we'll figure that out. So now, how do I switch to this one? Go back, leave. I'll let you drive.
Okay.
And I don't know whether we can actually increase the screen and maybe get the lights off. See if we can do that just for the video. No, that's you. That's You know where are these lights? Oh, we need to mute something. Not good.
From virology to immunology, communications to behavior to economy to the natural and built environment.
And create the platform from which all of these different people and entities can partner with solutions.
That is a lofty goal, but I think we're in the right place, and this is the right time to launch this consortium.
Here in Arizona, there's some remarkable things. Like reaching
Yeah, no, it's not going to work. We'll just go back to, we were talking about how we put together wastewater testing, which would give us an early indication of the virus being present in the dorms, together with antibody and PCR and antigen testing, and contact tracing with apps that would tell us who was with whom and when. And then we had like the whole system of isolators and of treatments, and how we were able, because we were centrally coordinated, to bring the campus back at the time when every other campus in the country was closed. Many universities tried to reopen, and they thought that our president was crazy when he said that we're going to reopen. Notre Dame reopened, had to reclose. UNC had to do it. We were successful. We brought, half a campus immediately, and then the other half in about 6 months. And because of that, we know what it takes. We know what it takes to, invent new tests and what it takes to scale up. But more importantly, this is really a human endeavor issue. We need to tackle communication. We need to tackle the economics. The whole false debate that we ended up in the country that ended up being so politicized. Is it health or economy? There's no reason why we would not have health and economy. In fact, if you want to have economy, you have to have health, and vice versa. And so there's a lot of things that we're really putting together. We can get back to the slides now so I can show you what we're talking about. So we call this the Aegis consortium, because Aegis is a word from Greek mythology, and it means something that confers physical protection. And in some of the, it's towards the end. Let's just scroll down. So, fourth, fifth, that one.
This one?
One, two above it. Yeah, that one. Perfect. Oops, that really didn't work. So my favorite meaning of the word is a protective cloak that Zeus and Athena were wearing. And we are hoping to provide such a cloak to the world in many ways. The fact that it's a consortium immediately tells you that it's not only our university, we mean it to be a convening space and a convening spot for other universities and industries and nonprofits and for-profits, and to really be this is what it is. And so the vision is a world prepared for any pandemic. And if we had Aegis in 2020, we would have had reduced sickness and death by proactive measures and advanced care. And we would have impacted communication and decision-making to better contain pandemic. And we would have provided solutions to protect both health and economy. That was interesting. So our purpose and the goals are to mitigate COVID-19 effects at all levels, because we're obviously not out of the pandemic yet, to use that knowledge to predict and prepare for the next pandemics. And that will include the impact of infectious pandemics on ongoing chronic pandemics. We have pandemics in our midst that are already here and are around all the time, like obesity, type 2 diabetes, even poverty and addiction, all of which are made worse by acute pandemics. This is looking absolutely horrible, but I'll tell you what it's supposed to mean. It's supposed to tell you that Aegis has three main pillars around which it is organized, and one is pandemic control, prediction, and preparedness. And we have our leaders on campus who are leading it. The middle one deals with chronic and long-term effects on pandemics, and not only from the standpoint of the infected person, but also from the standpoint of the caretakers, from the standpoint of the community, and from the standpoint of economy and what we do overall. How is this going to impact our learning going forward, our teaching, and so forth. And the third one is built in natural environments in pandemic control. So what do you do as you're building pandemic resilient? Can you make it that way? What kind of filtration do you have in the system? Can you know before you enter a building, how safe is it for you? So there were people put really cool apps that would tell you immediately, this is the building that has a capacity for X number of people based on the engineering controls and so forth. Currently has a little less than that. You should be cool to enter. So those types of things. And then we have these cross-cutting themes that are very important, that cut across pre-pandemic, mid-pandemic, and post-pandemic, and across all of our pillars, like the behavior before, during, and post-pandemic, in informatics and machine learning, policy and communications, food and water security. I mean, one of the things that, my lab rat brain never dealt with very well is like, how do you why are you not able to enforce the quarantine and social isolation? Well, you go to India and you've got people that have got no access to food. Obviously, they're not going to stay at home and isolate unless somebody provides that. I mean, those are just, this is highly, highly transdisciplinary. We have recruited the most brilliant people from our university, but we need a lot more. And we're really looking for partners, and are well on our way to find partners, within our government, within the state, within, the different industries, the government branches and so forth. And, wearables and engineering really important, including having a wearable that might predict whether you're developing long COVID right now or not. So that is the latest non-ambitious thing that we're trying to do.
Wow.
And I always get...
Every time they come and talk to us, I can't believe it. Well, see those other people.
Yeah. And I get the question, I get many questions about this, like, isn't this too ambitious? And my answer to this is that, my answer is always yes, because it is way too ambitious. It is ambitious beyond. But if we don't start addressing it now, we're going to be in the same shoes next time. And even if we fail at 90+% of it and we accomplish 10% of it or 5% of it, we'll be a lot better off. And we'll at least have a roadmap of what else needs to be done. So thank you very much.
Thank you.
I know we have some questions online, but we'll do it if anybody has any questions here, but wants to ask first.
Yeah, people here get priority.
I think so too.
Anybody here?
So the next pandemic, for instance, does that mean there will be immunization faster? Because each one has to be developed individually, right?
You're absolutely right. We don't know what the next pandemic will be. What we know is that, if you look at the last 20 plus years, we have had an outbreak of a mosquito-borne infection, which was West Nile virus here. It swept across the country in about 3-4 years. That's a different challenge relative to the respiratory. This one, in many ways, is the worst of what we can face. Respiratory infections typically are the most contagious for humans, and they give you the least time to react. But you are right, for everything that we do, part of the vaccine needs to be developed from scratch. The good thing is that if you have vaccine platforms that are, usable and scalable, then you can do it relatively quickly with modern technologies. That is why it was possible actually to develop these mRNA vaccines, because they were already researched for 15 plus years. It's not that we have done something really overnight. It is that we had something that was repurposed very efficiently. They were under investigation for cancer treatments for a long time already. So we knew about them. We had the platforms. All you had to do is to make the piece that corresponds to the virus and put it in there, scale it up, and then do all of the testing. So what was possible this time will surely be able to be reproduced next time in terms of speed. And I think in terms of improving the platforms in a variety of ways, well, hopefully we will do that as well. There are some measures for every pandemic that you can really literally make a playbook for and be ready and have them off the shelf. I mean, the first determination is going to be exactly that. Are we looking at the respiratory infection? Are we looking at something that is food and waterborne? Are we looking at something that might be mosquito-borne? And that gives you three broad categories already to set up, your measures. But vaccination is pretty much, you have to wait for about 9 months or so, and then we should have the vaccine for just about anything that we isolate.
How often pandemics occurring, or are we going to look at another one?
It is getting worse. Not only, yes. Sorry. The question was whether the pandemics are happening more frequently nowadays. Will there be another one in the next 20 years? Is it worse, perhaps because of travel? Certainly, the globe is more connected than ever in terms of travel. That's true. What is worse than that probably is that we keep encroaching on natural habitats that we weren't in before. And for instance, bats, who are great natural reservoirs of many viruses, emit a lot more viruses that they endogenously carry that are not harmful to them at all if they're under stress. Now, if you go with bulldozers and whack most of their natural habitat, they're going to be under stress, and they're going to fly into cities, and they're going to be close to humans. Not only that, the humans are going to be right there as they're trying to develop another piece. So that right there is another serious issue. The issue of these food and animal markets is enormous, because anytime that you put different critters together, everything that's in them will jump and try to see whether it can recombine and become better at infections. So if you look again at the last 20 years, I mentioned West Nile. We dodged the chikungunya epidemics, which was also mosquito-borne. The Caribbean did not. They had 1.3 million cases. And it's a very nasty disease. It gives you very bad arthritis that, again, unsurprisingly lingers a lot more in older adults. We had SARS and MERS that didn't move into pandemic stage ever, because the virus was relatively unsuccessful the way that it was designed, both of them. But they were both highly deadly. MERS is still killing about 25% of the people that it infects. So coronaviruses have been overall as a family pretty good, because you've got about more than half of them living in different animals, about 10% in humans, and then the rest in other non-vertebrates and so forth. Yes.
And then due to the severity of this particular strain,
I didn't, because Mike Warraby, who's one of the leading evolutionary biologists in the world and is also on our faculty at the university, published a really brilliant paper relatively recently in the journal Science about the spread of the infections in the city of Wuhan, that shows extremely clearly that the clusters of initial infection for a long while were around the market itself. They did. They found numerous animals that are known to be infected with these, and they have these animals in cages, one on top of the other, of different types of animals. That's a perfect laboratory to essentially get something like this. There was another opinion piece that you can read about. It was written by Bob Gallo, in the Time magazine about it, saying, to what extent this matters and to what extent this doesn't matter in terms of what we do next. That was published before that Science paper was published by Mike Worraby. But the evidence and the evolutionary evidence corroborating this right now is very strongly on the face that this was a natural infection and a naturally occurring virus.
Okay, Maya will come on with the online questions for you.
So I do have a couple of questions that came in. What is the status of research towards a development of maybe a nasal COVID-19 vaccine?
I don't know specifically where that stands. There were platforms, of course, that were, So the question was, what is the status of the development of a potential nasal vaccine? And again, it's an interesting point. I don't know whether we would want nasal or whether we would want oral, because Omicron, again, coming up was a lot more actually in the mouth than it was in the nose. You could get a swab in the nose and be negative and still have the virus in your oral cavity. Nasal vaccines were, certainly flu mist was distributed as a nasal vaccine before. It had some efficacy issues suggesting that whatever we had was perhaps not to be replicated again. But I know that, the research of this type continues. And so I just don't know what may be happening over the last 3 months right now.
Okay, and then I've got a question about chemotherapy and how that reduces the immunity. Does that happen years after, or can that happen for years after and make it harder to fight your viral infections? I do know, for instance, bone marrow transplants, typically if you've had one of those, you normally get you get revaccinated down the road, right? But I don't know how that works as far as the chemo piece.
Yeah, so the question was about chemotherapy and the relationship of chemotherapy to the regimen of vaccination. So just to go back to the basic regimen of vaccination, because of how this rolled out, we were believe that our third shot was a booster shot. And there's a little bit in the language here that we need to consider. Many vaccines that we get in childhood are actually a 3-dose vaccine regimen. And you typically vaccinate at the first point, then a month later, and then 6 months later. And this vaccine actually should be considered like that. You should consider your third shot being part of your original full vaccination regimen. Because, somewhat to my surprise, but not completely, there's really quite a bit of evidence that after the second dose in many people, there is a decline, particularly in the antibody levels. So with the third dose, I know that went up. We're still studying how long will that last and how much longevity will we gain from that third shot. But that now may become the topic of tinkering with the vaccine itself, because if 3 shots of this vaccine that don't give you something that's going to be lasting for about 2, 3, 4, 5 years or something like that, I think it's probably going to be a good idea to rethink, how that needs to be modified further.
So they're talking about new boosters.
That's
kind of a revised.
Yeah, so right. So anything after the third one should really be called a booster, and then we need to figure out what that should be. And should that be really then, Omicron based, for example, relative to something else? There's a lot of basic science and practical considerations that we need to answer there. Now, the question that you read from me, Maya, was about people on chemo and what's happening with them. There was a study that it was led by Rachna Shroff and Deepta Bhattacharya. It was collaboration of my department and the University of Arizona Cancer Center that looked in people with solid tumors, and it's essentially showed very nicely. That was at the time, again, when 2 doses were like your par for the course, and it was showing that most of the people on chemo with 2 shots had almost negligible immunity at that point, whereas the third shot was really important getting it up. And so in those people, the fourth shot may be something that would be a lot closer to reality and the reason to immediately, apply it. Whereas for most of the rest of the population, I don't see scientific reasons why the fourth shot should be discussed at this point yet. That doesn't mean that something may not happen that will make me reconsider that. We have seen in sporadic cases, because we are in contact with many of our patients. We had some that were immunosuppressed because of autoimmune conditions. And some of them were on pretty drastic treatments, for example, that would abrogate their B cell responses. Rituximab does that in certain, autoimmune conditions. That again, the third shot did wonders for the T cell immunity. The T cell immunity after the third shot in people that were profoundly immunosuppressed. Now, they're not going to make an antibody response no matter what, but they will have T cells that could protect them from severe disease. That actually came up to the realm of being within the range of normal responses that we would see in non-suppressed people. So that's a practical consideration that is important.
And then just one last comment. The person who was asking about the research on the intranasal vaccine was interested in the IgA mucosal antibody.
Well, that's exactly right. I mean, so the question was, that the follow-up question to mucosal immunization was a question about IgA antibodies. We have different flavors of antibodies, and they have specialized functions. And so IgA is the antibody that we make on the mucosal surfaces. It is the type that is ideally suited to patrol those sites and to be more resistant to. It's kind of like living almost outside of the organism, and it's not within the intracellular fluids. And that's exactly the type of antibody that we would want to generate by a mucosal vaccine. So that kind of goes one with the other. We know that, like when we detected after original infection, when we were looking at saliva in people who were infected with Wuhan, we could detect IgA antibody response. Now, how long were they lasting? And did the vaccines make any of that response that we didn't really study, although I guess we should. That's a relatively straightforward thing perhaps to check. But yes, that would be exactly the type of antibodies that we would want to generate with a mucosal vaccine.
So I just want to thank you so much for being here today with TMC for Seniors. You have such amazing information to share with us. So it was wonderful to have you. I want to thank our audience, and we will see everyone tomorrow. We do have another presentation tomorrow, and it is all about whether you have a hearing issue. So please stay tuned for that. That'll be on at 2 p.m. tomorrow. And thank you so much for being here, Dr. Nikolich.
Take care. Thank you.